Live streaming is on.
Okay. Good day to all listeners. Today's presentation were guided by Mr. Sagar Thakur Das from Nauraji Wadia College, discussing about the topic of biogenic and organo-sedimentary structures, presented by myself, Hamid Massri. The presentation is going to include two main topics to digest, which are the stromatolites and the trace fossils rates of deposition. Well, as you can see in the slide, starting with the stromatolites, we will be able to know first the definition, what is stromatolites, the distribution, where it can be found, formation, how it is arranged, and some figures for recognition. Moving to the next topic, which is the trace fossil rates, we are going to understand what do we mean by ichnology and the importance, the consideration of trace fossils, why we are studying it, some giving nomenclature for building a better relationship between trace fossils and environment, which will lead us to the correlation between depositional environments and the characteristic trace fossils assemblages termed as ichnology, ichnofacies. Okay, here are some questions to be asked. You might want to consider at the end of presentation. We'll be answering them within our presentation. What do you know about stromatolites, and what is the main difference between ichnology and body fossils? Okay. A broad definition for stromatolites can be given as below. First of all, stromatolites is considered as fossil. Fossil stromatolites are the oldest evidence for life on Earth. They first appear approximately 3.4 billion years ago, at the Precambrian time, in which it is a laminated structures composed of particulate sediments and or precipitated carbonate produced by the growth, metabolism, and the breakdown of successive algal films or crusts. On this particular figure, we can see that it is taking the shape of dome, while on the upcoming slides we're gonna see also some different forms for the stromatolites. Okay, distribution. As we said, we need to know where this fossil can be found. Well, algal laminated structures, termed stromatolites, are most prolific at the present day in very shallow waters of both marine and non-marine environments. In the marine environment, they are characteristic forms in the shallow subtidal to supertidal zones of tropical to subtropical carbonate environment. And that by itself means that there will be some difference. There will be a difference in the degree of wetting, frequency of exposure, and intensity of current actions. And by this effect, a variation of in stromatolites surface micromorphology occurs, so that we say that there is no general rule for the gradation. And now we're gonna be showing some algal structures, including some flat, smooth, crinkled, cinder, polygonal, dome, as we have seen before, and blister forms. Okay, here is our first figure, which is figure A, showing a smooth, regular, laminated algal stromatolites. We can observe here some dark and light variation within the figures. The light ones indicates for sediment rich, while the dark ones, the light one here, and the dark ones are algal rich, and it occurs in lower intertidal zones. And now for the second figure, figure B, it is a view of Leicester algal stromatolites from the mid-upper intertidal zone. Here we can observe the blisters. Moving forward, here in figure C, we can see the polygonal algal mat with raised rims here, as we can see it, and some areas in the surrounding covered by blisters mat growth. Here we can observe the rims, and here in the surrounding area we can see also the blisters. Here in figure D, we can see a section through the algal mat, which will be shown in a better manner with a video included with the presentation that gonna help us to clear out a lot of what we had talked about stromatolites. Well, figure E showing some lithified stromatolites. Let's say if you are really looking to check some stromatolites, then look further no more for an area called Shark Bay. It is in Western Australia. We're gonna be seeing it also with the video showing. Okay, now as figure F also showing lithified stromatolites ridges in exposed high energy intertidal zone, also from Shark Bay, Western Australia. It shows elongation parallel with the direction of wave propagation. Okay, now last but not least about the stromatolites, we're gonna be checking a video, as I mentioned before, showing how is it formed, the formation, and how it is arranged. And to be more clear, this video will include all of these points with details about internal structures and how it is solved, what is the dominant process, so that we're gonna skip on explaining them and paying attention to the video. Okay, I'll be sharing the video now. I hope it is visible for all.
You've ever seen. The Egyptian pyramids that are 4,600 years old, a dinosaur fossil that is 230 million years old. How about a stromatolite fossil that is 3.7 billion years old? What is a stromatolite? Stromatolites are layered rock-like structures made by the activities of microbes. To see living examples of the world's most ancient ecosystem, look no further than Shark Bay in Western Australia. Why should you care about stromatolites? Well, one of the most prevalent microbes in living stromatolites are cyanobacteria. Cyanobacteria are microbes that feed off the sun using a metabolism called photosynthesis, just like plants. One of the most important characteristics of cyanobacteria is they provide much of the oxygen we breathe. Because of these cyanobacteria, stromatolites have played an important role in creating the oxygen-rich atmosphere we have today. Also, stromatolites grow towards the sun, just as plants do. When you see the insides of a stromatolite, there can be thousands of individual layers, each layer building on top of the other. So the newest, youngest layer is always on the surface, with the microbes searching for the sun. So what are we going to do if these amazing stromatolite ecosystems ever die out? It's an important question, as living stromatolites are highly susceptible to climate change and rising sea levels. Not only do these amazing ecosystems help generate the oxygen in our atmosphere, but also living stromatolites represent an important window into the ancient Earth. Studying stromatolites helps us understand how life has adapted and evolved to the ever-changing Earth. Knowing more about our past helps guide us to understand and respond to present and future environmental issues. So if you're ever in Western Australia, be sure to stop by and gaze at these magnificent ecosystems.
Okay, I hope it was clear for all. Now, just give me a minute to come back for the presentation once again. I hope it is visible for all. Is it visible, Mr. Sagar?
Yes, it is.
Okay, sir. Okay, coming back. I know it might be a bit lengthy, but I guess for now, if by any way someone asked you about what do you know about stromatolites, you'll be having some idea. Moving forward to the second main topic, which is ichnology, or we can say trace fossils. Now, what is biogenic structure? A great variety of structures in sedimentary rocks can be attributed to the work of organisms. These structures referred to be as biogenic. Biogenic structure include plant rootlets, vertebrate footprints, trails, some soft sediment burrows, and hard rock borings. These phenomena are collectively known as trace fossils, and their study is referred to as ichnology. Biological fossil records can be divided into two main categories, which are body fossils and trace fossils. Body fossils and the trace fossil, and here we can see the answer of one of our questions at the beginning of the presentation, which is what is the main difference between ichnology and body fossils? Well, as we can see here, body fossils are nothing but preserved remains of the plant or animal itself, while the trace fossils, or we can say ichnology, may consist of impressions, which are behaviors made on or in the sediment by an organism. Here, as you can see, number 1, 2, 3, and 1, 2, and 3 is happening on the sediments at the upper section of this figure, while 4 and 5 is happening within the sediments at the lower section of the figure. But before explaining this, we need to know why should we consider trace fossils, why it is important for us to know. This information is important and for itself for reconstruction of ancient depositional environments. And how this approach can be achieved? Well, the shape of trace fossils reflects environments rather than create them. This means that trace fossils can be a very important indicator of the origin of the sediments in which they are found because of their close environmental control. Beyond any doubt, it can furnish valuable information concerning general depositional processes, episodes of local deposition and erosion, also characteristic of currents, substrate consistency, and in some instances, causes of sediment sorting. Okay, once again, as we said, in and on sediments, nomenclature for building a relationship. Sedimentologists need to know something about the trace fossils, some basic principles of occurrence and nomenclature will be described before analyzing the relationship between trace fossils and environment. These nomenclature, 1, 2, 3, as we said, happening on the surface, which are resting traces called as resting traces, crawling traces, and browsing traces. While 4 and 5, feeding structure and dwelling structure is happening within the sediments, as we can observe here. Further to clarify with this, okay, so first one is resting traces, which are nothing but marks left behind by organisms resting in one place. As we can see here, some resting traces made by seal coming to rest on sandy paths of tidal flats. Second one is crawling traces, which are marks made by organisms moving on soft sediments, as we can see here, like a snail, we can say for an example, crawling traces. Third one is browsing traces, which are marks made by organisms feeding on a sediment surface, as we can observe here, some browsing traces on the seafloor. The fourth one is happening within the sediments. Now we are moving to the 4 and 5, which are happening within the sediments, such as feeding structures, which are pores and other structure produced by organisms while moving through the sediments for feeding. Here we can see as well, well-defined spiral and meandering porosystem. And the fifth one is dwelling structure, which are porous and other structure made by organisms in which to live. Here we can see some porous may be backfilled, as we can see here, by dwelling structures. The animal is visible in the poro here, so it can be moving and feeding through the sediments. Okay, now moving to our last topic, which is ichnofaces. Ichnofaces, the correlation. Ichnofaces, the correlation. The most, we can say the most useful aspect of trace fossils is the broad correlation between the positional environment and characteristic trace fossils assemblages, termed as ichnofaces, which was drawn by a scientist called Selinger at 1964. And as you can pick out from this figure, he has divided it into four sections: terrestrial, first one terrestrial, intertidal, third section is subtidal, and deep marine. Deep marine. That we're gonna further clarify, starting with terrestrial, which is the most landward ichnofaces to be defined, consist here from the first section largely of vertebrate tracks that include the footprints of birds, as we can see here, and some terrestrial animals. Dinosaurs tracks, we can say that it is particularly well-studied examples, and the preservation potential of such tracks is low. They are most commonly found on dried up lake beds, river bottoms, and tidal flats. Also on some upper parts of alluvial fans, where the dinosaurs migrated to breed where the weather was cooler. The first section here is the terrestrial. Okay. Moving towards the sea. Moving towards the sea, well-defined ichnofaces occurs in tidal zone here, the second section. This is often named the Scolithus assemblages because it is dominated by deep vertical porous of ichnogenous Scolithus. Here, as we can see, the porous may be simple vertical tubes. Here, simple vertical tubes like Scolithus, and also some vertical U-tubes like Diplocraterian. Here. Okay, as for the third section, in the subtidal and shallow marine environment, Cruziana and Zoophycus ichnofaces, here are the Cruziana and Zoophycus ichnofaces, have been defined. Also invertebrates porous, as you can see here, invertebrates porous, as you can see here, it tends to be taking the shape shallower and more oriented obliquely or subhorizontal. The Cruziana ichnofaces referred as to be the action of trilobites, while the Zoophycus here, as we can see, it characterizes a helical spiral from in plain view, and it is generally present at sand shell interfaces. Finally, into the deep white water, a further characteristic ichnofaces is named after nereids. Here in the deep white water, in this environment, invertebrates live on rather than in the sediment substrate. Porous are largely absent and surface trays predominant. Characteristic meandrum forms traces include nereids, helmintida, and cosmohath, as we can see here, showing the shape of snakes, some like snakes, and some polygonal reticulate trails such as paleodietan are also characteristic of these ichnofiches. Here we can observe this one. The nereids ichnofiches is commonly found in interbedded turbidite sand shell sequence of flash faces. Thank you all, and hopefully that you enjoyed the video. Any further questions, we'll try to answer it with the comment section down below. Here are some references that it might be useful for you. I'll be stop sharing now, Mr. Sagar.